So I'm very excited to be introducing our first speaker of this morning's sessions on spatial imaging, Dr. George Emanuel. Right, yeah, it's up right now. So George is a scientific co-founder of Vizgen. So he did his PhD in biophysics from Harvard University. And over the years, he has been spending a lot of time developing high-throughput spatial profiling technologies, including extending the MERFISH technology to profile over 10,000 genes in the same sample. So I think this morning he'll be speaking to us from Boston. Then we'll be showcasing some of the amazing work that his team is working with the MERSCOPE. So take it away, George. Can you start sharing the screen?
Hello, yes, I just shared the slides. Are they coming through?
Yes, all good. Thank you, George.
Great, thanks. I'm excited to be here today, at least virtually, to present what we've been working on over the last couple years within Vizgen to help bring MERFISH technology into a commercial solution. I'll be discussing both MERFISH technology, introducing how the technology works, briefly describing how that has been turned into the commercial solution to help streamline the whole workflow, all the way from custom gene panels through data acquisition to data analysis. And then I'll close out in the second half with a number of different case studies where people have applied MERFISH technology to really advance their biological research. But overall, I'm really excited for this symposium because it does show this very quickly growing interest in spatial biology. In particular, this merger with techniques that are traditionally in the realm of genomics, where you have to extract RNA molecules, put them into a sequencer, count the sequences, number of each of the different RNA transcripts, in order to understand a genomic scale level of gene expression, while at the same time merging that now with things that are traditionally in the pathology side of the research arena. Now, with these technologies that are starting to emerge, including MERFISH with the MERSCOPE platform from Vizgen, we're starting to be able to measure gene expression at a multiplexing capacity that allows us to really understand what's happening, but now do that with the full spatial tissue context. Since all biological systems are very intricately spatially organized, this has the promise of opening up a whole new set of biological discoveries that now being able to measure gene expression at the level of hundreds or even thousands of genes, while knowing exactly where in the tissue each of those transcripts was detected, lets you really see exactly what's happening within that biological tissue and understand it much, much more better than an isolated system where you don't know where the cells are or where the transcripts are. So within Vizgen, we do that through the MERSCOPE platform, which is a combination of MERFISH, a highly multiplexed single molecule FISH assay, with an easy-to-use platform solution that we call MERSCOPE. MERSCOPE streamlines the whole workflow all the way from designing a custom gene panel, where since it's an RNA targeting approach, it's very easy for us to design new probes against new genes, and in that way construct just computationally panels for genes that are hundreds or even thousands of transcripts in size. So in this way, we're very easily able to design custom panels that really suit the biological question that you're looking into. Along with that, we also have reagents and consumables to help with sample prep. We have instrumentation that runs through the full MERFISH process that I'll be describing in more detail. And then we have downstream image processing and visualization software that helps users really understand what they're getting off the platform. So with MERSCOPE and with MERFISH technology, we're able to profile genes at a transcriptome relevant level of multiplexing, hundreds or even thousands of genes in a single instrument run with very high sensitivity and accuracy. And we can do this a lot across large tissue samples of up to a square centimeter of tissue, all with single molecule resolution across that whole square centimeter. And then with the MERSCOPE platform, since all of the imaging happens for the MERFISH readout within the instrument, there's no need for downstream sequencing. We combine this technology platform with easy-to-use tools that really are starting to enable different researchers to apply the technology across a whole range of different biological fields. So today, I'll start out by introducing MERFISH technology, how we're able to achieve this very highly multiplexed spatial transcriptomics measurement. From there, I'll describe briefly what the MERSCOPE workflow looks like, and then I'll close out with these few biological case studies where people have used MERFISH technology to understand what's happening within their biological tissue. So MERFISH is an acronym, as with a lot of different methods, but in this case, MERFISH stands for multiplexed error-robust fluorescence in situ hybridization. As the name suggests, it's built upon fluorescence in situ hybridization, or in particular, single molecule FISH. In a single molecule FISH measurement, you're able to very accurately characterize gene expression by designing fluorescently labeled probes that bind specifically to a target transcript. So that once these fluorescent probes are hybridized onto the sample, then you're able to look at the sample under a high resolution fluorescence microscope, and then each copy of the transcript will show up as an individual point under the high resolution microscope. So then just by counting the number of spots, you both can characterize the magnitude of the gene expression, but you also know exactly where each of those transcripts is. So by combining that with cell segmentation, then you count up the number of transcripts within each cell and see how gene expression is varying across the whole sample. So single molecule FISH is very highly quantitative, but as with a lot of traditional fluorescence techniques, it's limited in the multiplexing capacity, where in a traditional fluorescence measurement, just based on the spectral overlap of the different fluorescent dyes, you might be able to image 5 or sometimes 7 distinct colors within a single imaging round, which is much less than is needed in order to really understand what's happening across the full transcriptome. So MERFISH is able to greatly increase that multiplexing capacity by, rather than reading the genes out one at a time, we assign each of the genes a unique binary barcode, a unique series of zeros and ones, and then we read out that binary barcode using sequential rounds of single molecule FISH imaging. So once each of the genes is assigned this unique barcode, then we can stain a sample sequentially in a way that we're able to read out the barcode 1 bit at a time. So in the first round of imaging, we stain the sample in such a way that all of the genes that were assigned a barcode with 1 in the first bit position will show up as single molecule FISH spots, while all of the genes with a 0 in the first bit position remain dark. So we first image this pattern of single molecule FISH spots to read out the first bit, and then we extinguish the signal and then restain in order to read out the second bit. So for the second bit staining, we stain the sample so that any gene that was assigned a 1 in the second bit position will show up as a single molecule FISH spot, while other genes remain dark. So again, we image this single molecule FISH to read out the second bit, extinguish, and then continue this process in order to read out the full binary barcode. Once we have this stack of images, then we can go back and look at where there's brightness and where there's darkness at each position across the tissue. From that, we're able to identify the binary barcode. And since we know which gene was assigned to each binary barcode, this lets us identify which transcript is at each position across the tissue. So in this way, with this combinatorial labeling scheme, sequential imaging, and error-robust barcoding, we're able to maintain this quantitative accuracy of single molecule FISH while expanding the multiplexing capacity to hundreds, or demonstrating on the academic side, even thousands or tens of thousands of distinct RNA species that you can measure all within the same sample simultaneously. So MERFISH was originally demonstrated back in 2015 in Xiaowei's lab. Since then, there's been a number of developments of the technology, but we're very excited within Vizgen to start to get the technology into the hands of a variety of researchers ever since our US launch earlier this year and our international launch that's coming up in just a few more weeks. So with this technology, then you're able to look at a tissue slice, for example, this mouse brain chronoslice, and measure gene expression across the whole tissue slice with single molecule resolution, so we can zoom into any region of the tissue and really understand exactly what's happening. From the way the technology works, there's a couple of technical advantages I'd like to briefly highlight, starting with the cell throughput. Since it's imaging-based technology, in order to capture more cells, we just have to tile the XY stage across additional areas of the tissue. And in a single run, we can detect nearly 1 million cells across that centimeter square of tissue. Next, since MERFISH technology is built upon fluorescence in situ hybridization, and FISH has already been broadly applied across different systems, it's very straightforward to apply MERFISH to new tissue types or even new organisms. Next, since for MERFISH it's direct hybridization, we don't need to extract the RNA molecules or perform molecular biology in order to amplify the signal. But with this direct detection through hybridization, we're able to maintain very high sensitivity, which is particularly important when it comes to transcriptomics, where a lot of very functionally relevant genes are expressed at relatively few copies per cell. Next, MERFISH has very high resolution, and it has that single molecule resolution across the whole imaged area. And then finally, with this barcoding scheme, we have custom multiplexing capacity, where both we allow researchers to pick exactly which genes are most suitable for their biological question, but also have the ability to balance between gene throughput and sample throughput to achieve the right balance for each different biological question. So with these advantages with MERFISH, we're able to achieve very quantitative measurements of gene expression. Here we're just looking at the copy number per cell detected in a MERFISH measurement and comparing it to a match sample for bulk sequencing. We can find that we have linear relationship between the counts detected with either technology across nearly 4 orders of magnitude of gene expression. We're able to accurately measure anything from the highly expressed genes to the lowly expressed genes and everything in between very accurately. As I was mentioning, since it is also this direct hybridization-based approach, we're able to achieve very high detection efficiency and very high resolution. You can compare this briefly to technologies that do require extracting the RNA molecules, where maybe you have to either extract different individual cells or capture it onto an oligo array. Since these techniques introduce inefficiencies, oftentimes that ends up hiding some of the very relevant biology. Often, when fewer transcripts are detected, then it becomes very difficult to characterize both the spatial organization and the single-cell transcriptomics. With MERFISH technology within Vizgen, we work to build it out into a platform solution. So building on the technology coming from the lab at Harvard that invented the technology, we were able to build it into a solution that's much easier for research labs to start to adopt on their own. As I mentioned earlier, this solution contains everything from gene panel design through data analysis, but the process starts with custom gene panel design, where we have a web application that lets users specify which genes they'd like to include in their custom gene panel. It provides immediate feedback about which genes are most suitable for a MERFISH measurement, and lets the user very quickly converge on a finalized list of genes that then Vizgen is able to provide in a ready-to-use format. With that ready-to-use gene panel, then a user is able to take a tissue slice, hybridize the probes in the gene panel against that tissue slice, gel embed and clear to remove any autofluorescence background, and then load the sample into the MERSCOPE instrument. The MERSCOPE instrument performs the sequential imaging process that I was describing earlier, where we read out the bits in the binary barcode. It automatically processes these images in order to construct the biologically useful output, which is a list of all of the detected transcripts across the sample, which could approach 1 billion transcripts in a single instrument run. It also performs cell segmentation, From the cell segmentation, we can count how many transcripts appear within each of the cells and construct a cell by gene matrix that is very similar to the output that you get from a traditional single cell sequencing experiment, where you can use that cell by gene matrix to cluster cells, identify different cell types and states. But now, since it's a spatial measurement, you can map all of those cell types and states back to their spatial coordinates across the tissue and understand how the cellular distribution is varying across different areas of the tissue. To help with that downstream analysis, we also have interactive visualization software that displays all of these different output components, the transcripts, the cells, and the images. Of course, for any tool, the true value of it is really judged by the sort of research that it can help enable. So in the second half of the discussion this morning, I'll go through a few different case studies of MERFISH. But following both our US launch earlier this year and soon our international launch into Australia, one of the things I'm most excited about, seeing the technology all the way back from when it was in the academic lab in 2015, is just all of the different ways that people are starting to apply MERFISH technology to their own research. To help towards that direction within Vizgen, along with developing the platform, we've also spent a lot of time demonstrating MERFISH in a variety of different tissue types, including mouse and human. As you can see here, nearly 30 or more different tissue types. And for each of these different tissue types, it's very straightforward to apply MERFISH as long as the RNA quality is good within the sample. So the first case study that I want to discuss is this flagship MERFISH paper published back in 2018 in Science, towards the early days of MERFISH technology. But here, to demonstrate how MERFISH can work to map out the molecular and cellular distribution across tissues, they applied MERFISH to a region of the mouse brain called the hypothalamus. This region of the brain is known to be implicated in certain behaviors like parenting, aggression, and mating. And they wanted to use MERFISH to better understand how the cellular composition of this tissue and the structural organization helped contribute to these different behaviors. So in all, they measured about 1.5 million cells with MERFISH across the different conditions, about 500,000 of them in the control condition. But here from the t-SNE, you can see they were able to very accurately characterize all of the major cell types across the brain, but also very finely differentiate between the different types of inhibitory and excitatory neurons in a way that's very consistent with what they saw with single-cell RNA sequencing, where nearly all of the single-cell RNA sequencing clusters were recapitulated in the MERFISH measurement. There were even cases where MERFISH identified multiple clusters when only one cluster was visible or distinguishable with single-cell RNA sequencing. In these cases, they found that looking into it more, that these clusters were discriminated based on the expression of neuropeptides, which are very functionally relevant genes for this region of the brain, but also relatively lowly expressed, so not able to be accurately characterized from the single-cell RNA sequencing measurement, but detected with MERFISH and validated through other techniques. So in this way, even using a panel of 155 genes, in this case, they were able to detect cell types that are invisible to what is considered the standard for identifying cell types across tissues these days. But of course, since it's a spatial measurement, they can map out all of those cell types and their spatial organization across the volume of the MPOA region. And then by combining the measurement with measurement of an early activity reporter gene, CFOS, and exposing the mouse to different parenting, aggression, or mating behaviors, they were able to understand which subtypes of inhibitory and excitatory cells were activated or implicated for these different behaviors to help understand how this cellular map that they were able to build up corresponds with a functional map, how these cell types are working together to overall contribute towards these different behaviors. So this is one of the earlier demonstrations of MERFISH, but also I think a very exciting first biological demonstration of the power of the technique. More recently, there's been other demonstrations in the brain. For example, this study coming out of the Broad Institute where they wanted to profile GABA receptive microglia, in particular to understand better how these microglia were helping to prune GABAergic synapses during development. So here they designed a very small gene panel, 20 or 30 genes, that very targeted towards understanding microglia. They were able to identify the different microglia subpopulations that they identified with their single-cell RNA sequencing measurement, and then see how gene expression within those subpopulations varied under their knockout condition to better understand how these different subpopulations of microglia were contributing to the GABAergic synapse pruning. The next showcase I want to highlight is also from an early Vizgen user, in this case, Roy Chen at Baylor College of Medicine in Texas. Roy Chen is an expert on the retina and applied the MERSCOPE technique to measure these 368 marker genes across the mouse retina. So here we're looking at just a single field of view from the measurement. You can see all of the detected transcripts across the field of view. Overlaid on the DAPI, you can see all of the different layers that you'd expect. But then through combining it with cell segmentation, they can look at the gene expression composition of each of these cells, cluster the cells, and identify all of the different cell types that they were able to identify from a single cell RNA sequencing measurement. In addition to that, since it's spatial, they also wanted to look at how the retina structure was perturbed when they knocked out one of these genes that they knew was critical for the development of the retina, in this case, LHX3. When they knock out this gene, they can both look at the structural disruptions across the retina and also see which cellular populations were particularly perturbed. They saw depletion of both this BC1b cell type and this BC6 cell type. But beyond the mouse, beyond these examples of different healthy tissues, then of course there's applicability to disease tissues as well, in particular in oncology. Here is a work using MERFISH where they wanted to understand the interactions between the immune cells in glioblastoma and some of the other and the progression or transitions that were occurring in some of the other cancer cells. In particular, mesenchymal-like glioblastoma cells that they were able to identify through their single-cell sequencing measurement. Through the MERFISH panel that they derived, they were able to very accurately and consistently recapitulate and identify the cell types that were identified with the single-cell RNA sequencing measurement. And with the spatial information, they were able to better support the hypothesis that macrophages in the vicinity of these mesenchymal-like cells were actually helping to induce a mesenchymal-like transition. And in this way, better understand some of these interactions between the immune cells and the cancer cells in this glioblastoma model. But beyond fresh frozen, these fresh frozen examples that I went through, one of the things that we're all equally excited about within Vizgen is the applicability of MERFISH across FFPE tissues. In particular, FFPE has certain challenges because sometimes the RNA starts to degrade over time based on how the FFPE is stored. And also in the FFP sample, the RNA becomes very heavily cross-linked, so it might become more difficult to hybridize probes against that FFP sample. So over the last year or so, within Vizgen, we have been working to develop and are nearing release of FFP sample prep procedures. Just to show how consistent they are with the fresh frozen, we can look at this FFP mouse brain slice using a panel of 483 different genes. Here, when we compare the expression of key marker genes, they compare very well with what's expected based on the Allen in situ hybridization atlas. But we also find very good quantitative agreement between the FFP prepared sample and a sample prepared from a fresh frozen tissue block. First, for the FFP, we get, again, of course, get this good correlation with bulk sequencing. We get good correlation between replicates. And we also get very strong correlation between the FFPE prepared sample and a fresh frozen prepared sample, to the extent that when we cluster FFPE mouse brain measurement with a fresh frozen mouse brain measurement, the sample in the UMAP and in the clustering results, the clusters emerge based on their cell type identity rather than based on the method of preparation. So the FFP and fresh frozen cells cluster together based on their cell type identity, independent of the sample prep method, which I think is very exciting to see, since these are such distinguished sample prep methods where FFP you fix and embed in wax and go through a number of different steps, but at the end of the day, you're still measuring the same thing, whether it's a FFP or a fresh frozen sample. With that, I'd like to close out since I'm near the end of the time. But as I was mentioning, what I'm most excited about as we look forward towards the next few months is looking towards the first international placements of the MERSCOPE instrument following our US launch, and to start to see what researchers within Australia can start to do with the platforms and the sorts of discoveries that they'll be able to make.
Thank you, George, for a really wonderful talk on such impressive technologies. So for our in-person attendees, so I've got 2 microphones down the aisle, so please come up and ask the questions to George. And while we wait for in-person attendees, we might start with online questions. So there's a question from Sarah Ellis. So how long does it take to analyze a 1 square centimeters tissue per genes? And can the MERSCOPE handle tissue microarrays?
So for the instrument time, it takes about 1 day per sample. So during that day, you're able to image a 500 gene panel across nearly a square centimeter of tissue. So that square centimeter doesn't have to be a contiguous square centimeter, but it could also be a number of different small tissue sections arrayed on the MERSCOPE slide. So in that way, we have seen some users place, for example, 6 or sometimes close to 10 distinct samples onto a single MERSCOPE slide, which then counts as a single run on the instrument and would take about a day for this image processing step on the instrument.
And Rory?
George, thank you for that wonderful explanation. So this isn't so much a question as an announcement. So it's not something we've been trying to keep secret, but I can announce today that in about a month, we will be taking delivery of the first MERSCOPE in Australia and APAC. So the first one after the initial release. So this is an instrument that will be here for several months, I think, and we'll be using it to really evaluate the technology. So that's something else for people to come and talk to us and the distributors about.
Yep, it's very exciting times ahead. Like, we can't wait to get our hands on it. Are there any more questions from the floor?
Hi, great talk. I saw in one of the papers that you presented that you found about, I think, 800 transcripts per cell. And I was wondering, and I think that it was in a panel of about 400 or 500 genes. And I was wondering if that is a normal number that you expect, or is that quite low, and does it restrict the analysis?
So for a typical measurement, we do detect on the order of a few hundred transcripts per cell, which we find is more than enough or at least enough in order to identify all of the different cell types based on their gene expression. Because often the panel that users pick, it includes a lot of the key marker genes. So then the genes that we detect are particularly informative about the cellular state. So there, with this few hundred genes, we do find that it is sufficient to characterize the cellular composition of these different tissues.
Thanks.
And considering the time, I'll just ask one final question from online, from Holly Anderton. So are there any tissue types that you have tried that didn't work so well. Have you tried skin tissue?
We have tried skin tissue, in particular with our FFPE protocol. The main challenge for a new tissue type is just the RNA quality. Since we are working to detect the RNA, if the RNA has started to degrade for one reason or another, then it becomes much more challenging to detect it accurately, and there can be a drop in detection efficiency. But one of the things that's also very exciting about the FFP protocol is that with this initial fixation step in FFP, it seems like it preserves the RNA a lot better than some fresh frozen procedures. Because with fresh frozen, it's just frozen. There's nothing preventing the RNA from degrading all the way through slicing the tissue, where the tissue thaws. All the RNases are still diffusing around in the time as that tissue thaws. And we think that procedure, since it's not as well held in place as an FFP, does tend to degrade some of the fresh frozen samples. So with these considerations, the main aspect is just the RNA integrity. But if there is good RNA integrity, then we do find that we can pretty reliably get a good MERFISH measurement from that sample.
Thank you, George, once again for a really wonderful talk.
Thank you.
Thank you.